Well, thank you very much. That was very embarrassing. I'm very glad to be here to speak to this audience. It's maybe a little different audience that I've spoken to before, but I echo actually what you said in your talk about collaborations and working together and working openly. And what I'm going to share with you this morning is just a few examples of how I got lucky when I finished my degree at Stanford. I'd been working in the lab and working on fun microbes, and I thought, well, is this really used in real life? And so I actually ended up working in consulting right after I finished my PhD. Started working with some great people in Ontario at Beak Consultants at the time, and from there got some insight into some really important problems, and we've been working together ever since. And so, working and sharing ideas is actually how we have to solve some of these problems, for sure. So I got interested. Let me make sure I'm doing this right. Okay. So I'm going to talk about what I've done actually for the last 20 years of my life in the lab is put dirt in a bottle, and it really is true. And then go after activity, function. If something works, then you can use it. And so the problem that has been vexing me and a lot, around the world is water. Contamination of water, particularly groundwater. So the group I was with at Stanford was a groundwater group, and it had hydrogeologists and geologists and chemical engineers, civil engineers, biologists, microbiologists, chemists, everybody together. Some pollution is very visible, but hydrocarbon contamination is pervasive. The nice thing about hydrocarbons is they've been around for a very, very long time. There's lots of microorganisms that degrade these, and actually bioremediation is the most common method for remediation for hydrocarbons on the planet. When they're in the subsurface, it's a little more tricky because they don't float on the surface and they don't mix so well. So groundwater remediation, the challenges there are absolutely mixing. How do you inject? How do you deliver anything to clean up a contaminated plume? So this image here is for organic pollutants that might be lighter, like gasoline or fuel, and that when they hit the water table would pancake out, but you still get this dissolved plume and contamination that you have to deal with. So over the last 20 years, I've been very much involved in organic contamination. Lately, we've been getting more into minerals, and I'll talk about that at the end, but the examples will come from this sector. So I've worked actually a lot on chlorinated solvents because chlorinated solvents, when I started working in the industry 20 or more years ago, that was the driver for cleanup at so many sites. And trichloroethylene and tetrachloroethylene are the #1 organic contaminants in industrialized regions, and the challenges with those is that they're more dense than water, and so when they get into the subsurface, they sink down and pancake out on lower permeability zones and go down further and far, and it's really, really challenging to clean up. But the neat thing is that these subsurface environments are really not sterile, and there's a lot of microorganisms within there, a lot of surfaces and interactions between the minerals and the microbes and the contaminants that brings about transformation. And so the fates of contaminants in the environment is actually not, is usually they do slowly degrade no matter what, no matter even if you don't even try. And so this is a gratuitous, pretty slide, really just to say that microorganisms are everywhere and they have a huge diversity, an absolute enormous diversity. And if we can tap in to some of the metabolic potential, the chemical reactions that these microbes do, there's the sky's the limit. And so just as a microbiology 101, we need to just think about what is it that microbes do, and what is it that all cells do? What is it that we do? What do we need? We need some source of energy to live, and you get that energy from chemical bonds. And so you need something called an electron donor, and we eat McDonald's hamburgers and we breathe oxygen, and the electrons get transferred from the McDonald's hamburger to the oxygen, and we expire CO2 and water vapor as we oxidize our electron acceptor and reduce reduce our electron acceptor, and to water. And so organisms can do this, we do this, organisms can do this, but they're actually much more clever than we, and microorganisms can use all kinds of substrates, benzene and toluene, even hydrogen as electron donors. Those get oxidized. They can use all kinds of electron acceptors in their metabolism. They can breathe nitrate and sulfate and iron, iron-3, any kind of oxides, and CO2. A process that I work on a lot is, is CO2. I knew I would get messed up with this machine. CO2 to methane, so methanogenesis, where this is just respiration of CO2 that gets reduced to methane. And anaerobic digestion is an actual amazing process, generates energy, and we really should be using it in our wastewater treatment processes way, way more than we are. In Europe, they're way ahead of us in anaerobic digestion. And I bet there's even areas in the mineral processing industry where anaerobic digestion could be used. So when you have a contamination in the subsurface, then if there's oxygen in the groundwater, the oxygen, so the white is oxygen. If there's oxygen in the groundwater, that oxygen will get rapidly depleted. Reoxygenation doesn't happen very fast. And then the next electron acceptor, maybe nitrate will get used, and then manganese or iron or sulfate. And ultimately, when all external electron acceptors are depleted, you'll be left with kind of a methanogenic or an anaerobic, very deeply anaerobic, low redox core. And this is what happens in the subsurface. And I've done a lot of work. You can sample and take dirt from any one of these redox zones and try to understand the microbes that set up along that redox gradient. And each kind of set of microbes is going to have different properties. And it's a really lot of fun to look at what potential they are, or what they can do. So I've spent a lot of time over the last year just taking samples from various places in the subsurface in conjunction with my colleagues in consulting companies and working at sites, to look at what are the microbes that are stuck to those sediments and what can they do with the contaminants that we show them. And we do this anaerobically because many microorganisms, actually oxygen is lethal to them. Methanogens, for example, that produce methane, they are really, really sensitive to oxygen. When they grow in an anaerobic digester, they're covered by other microbes that are facultative aerobes, and so the oxygen, they're protected a little bit from the oxygen in a community. And so we do all this in these glove boxes. This is Lena here, put her hands in a glove box, in a glove bag, sorry, and we prepare all these kinds of bottles under different treatments. We add nitrate or sulfate, or we don't add anything, and we see, and we wait over time, and we measure. And for a microbe, from a microbe's perspective, all that matters is that there's energy that can be derived from the electron donor and electron acceptor couple. And this is an example with toluene. So toluene degrades aerobically really easily. Okay? And it also actually degrades anaerobically really well. You can convert toluene to methane, okay? And it's not a problem as long as there's a negative sign here on the free energy overall of oxidation of toluene to CO2 coupled to the reduction of CO2 to methane, actually. It actually can happen. And we have studied this process in the laboratory. And what happens in toluene fermentation, basically, and conversion to methane and CO2 is you have a whole clades of different microorganisms involved in the transformation. So you have fermenting organisms that take toluene part of the way to fatty acids and alcohols, and especially hydrogen. And then that hydrogen is the wonderful substrate for methanogenic microorganisms. And this kind of food web, food chain, and microbial community is the norm. You don't normally have a single microorganism that does everything. It's a community of microorganisms that work really syntrophically, is the word, together. So it's syntrophically because these methanogens that consume hydrogen, what they do is they bring the hydrogen concentrations down to such low concentrations that they drive otherwise thermodynamically unfavorable reactions for the fermentation of toluene. So toluene wouldn't normally form hydrogen unless that hydrogen is sucked away, okay? And so the methanogens serve that role in the environment, which is really a fascinating process. We've been studying also a lot of, studying also benzene, and toluene and other hydrocarbons, and they all can degrade this way. It's a little slower than the aerobic case, but groundwater moves slowly, and so it really can significantly affect the fate of the contaminants in the water, nonetheless. So the other problem that I've worked on very extensively is the chlorinated solvent problem, and these are just such prevalent contaminants around the globe. And when I started working on this, a long time ago, this is what was known, is that you could go out there and measure TCE in groundwater, And what you'd see is a TCE, and you'd also see daughter products, dichloroethene and monochloroethene, also vinyl chloride. And this was a really bad thing because vinyl chloride is a proven human carcinogen and was just made the whole problem worse, okay? And so it was really thought that you don't want your TCE in anaerobic water because they knew this happened if you had no oxygen in the water. With oxygen, these compounds don't react, so they don't do anything in the presence of oxygen. In 1989, some researchers at Cornell University were studying the Ithaca sewage treatment plant and came up with, they realized that you could actually completely dechlorinate vinyl chloride to ethene. And this just unblocked a whole, the next 20 years of research to try and understand if we can really dechlorinate and remove the last chlorine atom off these molecules, then we have a really promising technology. And so at that time, I did this. I was working in consulting, just transitioning back into an academic position. We got dirt and put it from a site in Ontario that was just almost bubbling ethene out of the water, and stuck it in bottles. And within a few weeks, we had a complete conversion of trichloroethene via the intermediate cis, vinyl chloride, all the way to ethene in stoichiometric conversion here. This is total moles in the bottle over time. And this was remarkable. My colleagues in consulting were over the moon, and we worked together. And over the time, what we realized is what we were studying, and this has taken many years, I'm fast forwarding, it was a new way of life. It's actually the organisms that were responsible for this transformation actually use these chlorinated solvents just like we use oxygen in our metabolism. They use them as their terminal electron acceptor. So they reduce the chlorinated solvent. So perchloroethene is reduced to tri to di to mono or vinyl chloride, and vinyl chloride to ethene. Each of those is a 2-electron transfer step. They're reductions. The electrons come from hydrogen primarily in these organisms. And so a whole a slew of research was carried out to try and understand what are the microbes that actually carry out this reductive dechlorination. And the term organohalide respiration has arisen because they actually use these in their metabolism to generate energy and ATP in their cells. And then the hunt was on as what are these microbes? And the first one that was isolated is this one here that I am so sorry. I knew I would do this. This first one here called Dihilobacter restrictus, all it can grow on is hydrogen as the electron donor and PCE as the electron acceptor. And it's really most remarkable microbe. The really ones that really came out in this field to be so important are these organisms called Dihilococcoides that catalyze this critical detoxification step at the end here. And again, these organisms fit exactly in that food web I told you about, the methanogenic food web. Here we have any kind of fermentable substrate producing the hydrogen that these organisms like Dihilococcoides and others can use to dechlorinate the chlorinated solvents. And it's a community of microorganisms that works well. If you get them in isolation, they grow very, very poorly. And so in 2002, my colleagues in consulting, Dave Major, Evan Cox, and others, decided to form a company called Serum to market the microorganism to inoculate sites with the Dihilococcoides to bring about complete transformation of that vinyl chloride. So you wouldn't get vinyl chloride building up in the environment. And the company is still going very strong. They have probably 20 people. And this is the critter at the bottom here, Dihilococcoides. This is a more normal-looking micrograph of microorganisms. The Dihilococcoides are so small, the diameter here is less than 500 nanometers. It's the dots you can't see. The other things are the fermenting organisms producing the hydrogen, and this is the organism that is doing the dechlorination. And as a result of genomics and molecular biology, we have DNA tracking to be able to track these organisms. Once you inject them in the ground, you can follow where they go. And so that's what this gene track is about. And so Serum has done many such inoculations at contaminated sites. Many sites have now, this has been a decade now, so many sites actually have closed. This is what happens. You put it in a big tank. You have to keep oxygen away, remember, so it looks a bit like a beer keg. You take this beer keg to the field, you use a tank of argon, and you pump it down the well. They have production of over 1,000 liters of culture. Remember, anaerobically growing on chlorinated solvents, so it's a health and safety kind of concern. And they have worked in numerous places around the world now, and their map of work is, and we're continuing to work with them, going back to these field sites, tracking where the microorganisms have established, what happens post bioaugmentation, post remediation, what does the microbial population do? And the genomics, the science that's gone along has been phenomenal. The organisms have these tiny, tiny genomes, which is the sum of all the DNA within them. And in these genomes, they have maybe 30 different enzymes that code for those dechlorination steps. And we're trying to figure out what each of these enzymes does. And what we figured out a little bit. There's individual enzymes that catalyze the different steps in the dechlorination. And some organisms have enzymes that do the first part, and some have enzymes that do the bottom part. So now we have molecular tools to look for this enzyme here. And it's so fun to go to a conference and all these engineers are around and they're all talking about dihelicocoides and vinyl chloride reductase and all this kind of stuff, and they're all excited about it. And so, you know, looking at all the genome sequences out there, we know there's so many of these enzymes, which are the catalysts that break these things down, and they can do all kinds of different molecules. And so I'm working now with some colleagues in the Caribbean, because it's also fun to go there, and, but they have contaminated with pesticides that are on their banana plantations. And these are, look at this molecule here, chlordecone. It's been banned for a long time because it's so toxic, but it has residue still in these soils. And it has 10 chlorine atoms on it. And we have now these bottles, dirt in a bottle from Guadeloupe, and we're seeing dechlorination of chlordecone. And so these organohalide-respiring microorganisms have really led to a successful environmental biotechnology. Serum is not the only company. There's many around now that sell culture just to inoculate groundwater that has chlorinated organics in it, because it really just does work. And it works because these organisms, that's all they do. That's all they can breathe, the chlorinated organics. They don't do anything else. And so when you add them in there, that's what they do. So it's been really a remarkable journey. So just to close, I hope I don't know if I have enough time to, in the last 3 years, in conjunction with a hydrometallurgist at the University of Toronto, Vladimiros Papangalakis, who told me that, you know, well, they're having so much trouble with, well, with maybe we could use some of these waste ores that still have a little bit of nickel in them. Maybe would be a bioleaching possibility for those, much like there's bioleaching already for gold, or there's bioleaching for copper to some extent. And so the thought was, well, could we do the same thing? And so in the bioleaching process I'm learning about, but again, it involves some really interesting microorganisms, organisms that oxidize, that use the electrons in iron 2, organisms that oxidize sulfide from the minerals, and also elemental sulfur to sulfate. Some of these are the problematic organisms that create acid rock drainage and so on. And so the interesting thing about these microbes is that they thrive at low pH, some of them, and so on. So we started to look at this problem. And the problem statement was, well, could we recover nickel? Could we recover nickel from pyrrhotite or even pentlandite or some of these kinds of, and waste the sort of waste tailings that still have a little bit of nickel in them? And so we initiated 2 bioleaching projects, one with pyrrhotite, and this would be an acidic bioleach, to see if we could extract nickel. And the second one was, because I said, well, maybe we could do something anaerobically. Maybe, you know, with oxygen, when you oxidize these iron sulfide minerals with oxygen, you produce a lot of acid. If you use nitrate instead of oxygen, you don't actually produce, nitrate consumes protons, and so you don't actually produce quite as much acid. So that might be possible. Would you be able to bioleach at neutral pH? Is this a possibility? And so we've set up the same idea. Well, before this, you know, the microorganisms that we're trying to enrich from these sites would be using iron 2. They would be using H2S, elemental sulfur, iron sulfide, and they can use these solids. And these get oxidized to iron 3 or elemental sulfur, or from elemental sulfur all the way to sulfate, and all the sulfur species in between, actually. And you can use oxygen, but actually you can also use nitrate. As long as there's a negative delta G, as long as the free energy is there, the organisms can grow. And so we just did the same thing. We took water samples from, in this case, an acidic mine tailings, put them in bottles, incubated over time. Eventually, we enriched on pyrrhotite tails and transferred and tried to increase the solids loading to try and get something that would be more significant. And to date, we do have some pretty good, I think anyway, pretty promising results. So what this graph shows is percent of nickel. Let's say the black line is nickel, percent nickel recovered in solution over time. In the case where we've inoculated with microorganisms enriched sulfur and iron sulfide and iron and sulfide oxidizing microorganisms under aerobic conditions at low pH versus no microorganisms. And so there is considerably more nickel that's being released into solution. So it has some promise. Again, of course, we have to look at the economics and how you'd scale it up and the constant. So what we're doing now is trying to get these microbes to really like to grow in high solids concentrations, so it could be relevant for the field. We also want to look at neutral pH, and here there's these microorganisms called Thiobacillus denitrificans, and so they use nitrate as their electron acceptor. They can also use oxygen, and they'll oxidize iron 2 to iron 3. They'll oxidize sulfides to sulfate. But again, if you couple this with, say, something like pentlandite, it'll actually consume protons, not produce protons. So it'll actually not cause any kind of acidification. And so we want to see if this can happen. So we did exactly the same thing. We went out to a mine site. I tried to find somewhere. So what you want to find when you do these microbial prospecting like this is somewhere where the microbes and the conditions you're looking for has been around for a long, long time, and evolution could have happened. So, because microbes evolve relatively quickly, so if you can find a site that might have had some nitrate there, but also had this minerals there, and the right conditions, and those microorganisms will have adapted to that environment. And so I was kind of looking at where agricultural runoff that might have, brought some nitrate might be coexisting with where some mineral mining tailings might be. And so we looked at some surface waters, put them in bottles, and incubated a whole host, lots and lots and lots of bottles. And so the rate of nickel evolution, and also you have to play with, you have to monitor the pH carefully because of nickel solubility and pH and so on. But we are seeing nickel evolution coming out of this, nitrate reduction, a lot of sulfide oxidation, even with mineral tailings from pentlandite. And so I think there's a lot of room for finding microorganisms that might be somewhat selective. The bioleaching challenges are tremendous, and we need many people to put their heads together to have a look at this problem. But if you could have a win-win where you could use a tailings waste material and extract some valuable product you could sell, at the same time clean up the acid mine drainage problem, I mean, wouldn't that be awesome? It's a pretty incentive, a good driver. There has to be a sweet spot in pH, where you can selectively enhance nickel and not have all the magnesium dissolve or something like that, and maybe we can find these things. But it'll only be when we put our heads together. So I'd like to acknowledge everybody who's contributed to all of this work. It's a lot of fun to learn new things, to move from the organic contaminants into these metals and minerals has been a lot of fun. So I'm indebted to Vladimiros Papanglakis, who's the hydrometallurgist, and Vale, and also Glencore, and even Estrata we've worked with as well, and all the funding agencies for this work. And the work on the organic contaminants, a whole host of students over the years, funding from Canadian organizations like NSERC, but also from the US Department of Defense, the Strategic Environmental Resource Defense Program, which actually funds a large amount of remediation in the US because there's so many military bases that need to be cleaned up. The Ontario Genomics Institute, Genome Canada, have been instrumental in funding some of the science that's gone along in parallel with the development of the technology, which has been a great time. And I've been working in collaboration with the people at Syntrophic, Dave Major and Evan Cox and Michelle and others, and also with the spin-off serum. Actually, there are some of the students from my lab that started that spin-off. So it's all been a lot of fun, and I'll leave you with this picture, which is absolutely my favorite picture from a microbiology textbook, but for a bunch of geologists, maybe it's a nice picture as well. You know, what it depicts is the whole history of the planet in a 24-hour clock. And what it shows is that life first appeared on the planet maybe 3.8 billion years ago in the form of microorganisms. And for all of this period up until maybe 0.9 billion years ago, the only things that were on the planet were single-celled microorganisms, and that diversity that I talked about exploring every single niche. And in the early planet was anoxic, and so these microbes that can use redox couples in these anaerobic environments are actually some of the most ancient and most evolved and most adapted to these conditions. So we could find them. We should find them. So with that, I will close. Thank you.